Hello, everyone, and welcome to today's webinar, an overview of primary immune deficiency disorder, PID, and laboratory testing for PID. I'm Alexis Krause of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Binding Site. To learn more about our sponsor, please visit www.bindingsite.com. Now, let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I now present today's speaker, Dr. Vijaya Knight, Associate Professor, Department of Pediatrics, University of Colorado School of Medicine, and Colorado Children's Hospital. Dr. Knight graduated from Mysore Medical College, University of Mysore, India, followed by a residency in Neuromicrobiology and Immunology, National Institutes for Mental Health and Neurosciences, India. She then obtained a PhD in Immunology from Melbourne University, Australia, followed by 3 years of postdoctoral work at the University of California, San Francisco, studying immune evasion by tuberculosis in human macrophages. She then spent 2 years as a Juvenile Diabetes Research Foundation Fellow at UCSF, working on T-cell regulation in models of autoimmune disease. Dr. Knight then transitioned to the biotechnology industry and spent a couple of years as a translational research scientist and was involved in biomarker discovery and development and immune monitoring for clinical trials. Dr. Knight has, for the past 10 years, directed the Diagnostic Immunology and Flow Cytometry Laboratory at the National Jewish Health and is currently heading up Diagnostic and Translational Immunology at Children's Hospital, Colorado. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Dr. Knight, you may now begin your presentation.
Thank you, Alexis, and thank you to LabRoots and to Binding Site for hosting this educational section on primary immunodeficiency and laboratory testing. So the objectives of this seminar are to provide some definitions around primary immunodeficiency, the current classification for primary immunodeficiency. We'll talk about the laboratory diagnostic methods that are broadly used for primary immunodeficiency disorders, and I will go through some examples of laboratory analyses for specific immune deficiencies. So what is primary immunodeficiency? It's generally defined as a defect of the immune system resulting from a mutation in a single gene, and this may be inherited or sometimes could be a de novo mutation, indicating that it may arise later in life. These mutations may compromise development or function of a component of the immune system. Primary immunodeficiency results in increased susceptibility to infections, but may also present as autoimmunity, increased inflammation, and in some cases, increased susceptibility to certain forms of cancer. A hallmark of primary immunodeficiency is recurrent, persistent, and difficult to treat infections, and when a patient presents with these difficult infections, it should prompt the clinician to think about primary immunodeficiency and investigate regardless of the age of the patient. So let's take a quick overview of the immune system. The cells of the immune system all start from a hemopoietic progenitor cell, or a hemopoietic precursor, as shown in this figure, in the bone marrow. Now, during development, the precursor cells can take one of two paths. They may take the path of the lymphoid progenitor, which then results in the development of B cells and T cells, as well as natural killer cells, or they may take the pathway of a common myeloid progenitor, which gives rise to cells of myeloid origin, the monocyte and the granulocytes. Now, when we take a look at B cells are antibody-producing cells, and they may either develop into plasmablasts, which are otherwise known as the antibody factory and actually produce antibodies that we find in circulation or immunoglobulins, or they may develop into long-lasting memory B cells, which can recall a response to a previously encountered infection or an antigen. T cells may develop commonly into helper T cells or CD4 T cells, or cytotoxic T cells, CD8 T cells. Of course, this is a rather simplistic picture of development of the immune system. T cells have many, many more subsets, as do B cells, but these are the major subsets we often consider when starting an initial workup for immune deficiency. On the myeloid side of development, the cells may develop into monocytes, which can then develop into macrophages or dendritic cells. Now, these two particular cells are very important for innate immune responses, or once a pathogen is encountered, to be able to engulf it and present it to T and B cells in order to stimulate a memory response. The myeloid progenitor may also develop into the granulocyte family of immune cells that includes the neutrophil, which is commonly the first line of defense against bacterial and fungal infection, the eosinophil, which is commonly found to have a role in infections to parasites, the basophil and the mast cell, which are important mostly in allergic responses. So given the complexity of development of the immune system, a genetic defect anywhere in this pathway can result in either lack of development of a particular cell type or affect the function of one or more of these subsets. And looking at the pathway of development, it's easy to note that genes that are involved in the progenitor stage can involve multiple cell types, whereas genes that are specific to a T cell or B cell, if mutations occur in these genes, then those particular subsets may be affected. And again, the genes in the earlier part of development may result in a lack of these cell types in the periphery, whereas genes involved in the later part of development, as in differentiation of the T cell into CD4 or CD8, may result in a functional impairment of these cells. So how does immune deficiency present? Now, the Jeffrey Modell Foundation, that has been involved in advocacy for primary immunodeficiency, has these posters that you may find when traveling around the country in doctors' offices, and they're used very heavily to educate patients on the signs of immune deficiency. So let's take a look at the 10 signs of immune deficiency, clinical signs and symptoms that should point towards immune deficiency. From the poster on the left-hand side of this slide, we see that if the patient presents with 4 or more new ear infections within a year, 2 or more serious sinus infections within a year, 2 or more months on antibiotics with little effect, 2 or more pneumonias within a year, a failure of an infant to gain weight or to grow normally, recurrent deep skin or organ abscesses, persistent thrush, which is candidial infection in the mouth or fungal infection on the skin, the need for intravenous antibiotics to clear infections, or 2 or more deep-seated infections, including septicemia, and also a family history of primary immunodeficiency. So patients presenting with any of these warning signs would warrant further investigation into their immune system to determine if there are any abnormalities that might point towards primary immunodeficiency. The poster on the right presents this information in a more child-friendly manner, because as we are aware, primary immunodeficiency we tend to find quite commonly in pediatric patients. So the same information is presented in a series of cartoons, making it easier to explain them to our pediatric patients. Now, having said that primary immunodeficiency manifests in the pediatric age group, it's always been considered that these are fairly rare immune deficiencies. These are fairly rare clinical conditions. However, over the course of the years, we have found that primary immunodeficiency is probably more common than previously thought. So this slide is taken from a paper published in 2014, and it shows the 10-year trend of patients that were followed in clinics, that were identified with primary immunodeficiencies, patients who were referred to centers where patients with immune deficiencies are treated, or patients receiving intravenous immunoglobulin. And as you can see here in all of the graphs, in 2004, these numbers were fairly low in the 5,000s. And as you can see over the years, there are certainly larger numbers of patients who are being treated, identified, and followed. And it's not because the incidence of primary immunodeficiency is increasing, it's because we have better tools to recognize them both clinically and within the laboratory. So the little cartoon that you see up here on the right that says Think Zebra, which has been used by the Immune Deficiency Foundation, is really to say, formerly in medical school, we're all taught that when you hear hoofbeats, think of horses, or think of the most common clinical condition, the likeliest possibility. However, for immune deficiency, it's important to say, well, when you see a patient with chronic infections, perhaps we should be thinking of zebras. Think about immune deficiency disorders that, though they may be rare, are being recognized increasingly just because we now have a better sense of both immune deficiency and laboratory tools for diagnosis. So the clinical presentation of immune deficiency can be variable. We just discussed the fact that patients often present with multiple infections, difficult to treat infections. However, the clinical presentation can be variable. Now, in the case of chronic granulomatous disease that we will discuss a little later in this presentation, this is a disorder of the neutrophil population specifically and can be inherited in an X-linked manner, which means it presents mainly in baby boys. So in a clinical presentation of this sort where a baby boy soon after birth presents with serious bacterial infections, it may be easier for the clinician to say, well, let's look at neutrophil defects since it's pointing us that way. Common variable immune deficiency, on the other hand, is a very heterogeneous presentation. It can present at any age. Patients may present with recurrent infections. They can present with chronic lung disease, with gastrointestinal disease, autoimmune disease, or even increased incidence of lymphoma. So CVID, or common variable immune deficiency, is sometimes thought of as a catch-all bucket for immune deficiency, and the presentation can be highly variable. Autosomal recessive chronic granulomatous disease doesn't follow the pattern of X-linked chronic granulomatous disease. It may present in males or females and may present later in life with milder infections. And finally, even though we consider primary immunodeficiency to present mainly as infections, we're now aware that the clinical presentation may be autoimmune rather than infectious. So given this wide variability of presentations in these patients, it's important that we do consider immune deficiency and investigate either to rule in or rule out. So primary immunodeficiency has been studied for many, many years and classified and reclassified as we have learned more about these diseases. The IUIS is the International Union of Immunological Societies, and within the IUIS, the Primary Immunodeficiency Expert Committee has published every year since the 1990s a classification of immune deficiencies. And so as we've learned more about these immunodeficiencies, we've moved from classifying them simply into T-cell, B-cell, or neutrophil deficiencies into a broader classification that has allowed us not only to investigate these patients better, but also to provide good clinical information. The latest IUIS classification was published last year in 2017 and classifies immune deficiencies into 9 different groups. The first one is a combined T and B cell immune deficiency. The severest form of this category is SCID, or severe combined immune deficiency, which we will discuss in a little more detail. The second category contains well-defined immune deficiency syndromes. An example is hyper-IgE syndrome or Job syndrome. The third category, predominantly antibody deficiencies, and the prototypic antibody deficiency is X-linked agamma-globulinemia that we will touch upon. The fourth category contains diseases of immune dysregulation, and an example here is autoimmune lymphoproliferative syndrome. The fifth category is congenital defects of phagocytes, so chronic granulomatous disease, which we discussed a few minutes ago and will touch upon under laboratory diagnostic strategies. The sixth category, defects in intrinsic and innate immunity. The seventh, autoinflammatory disorders. The eighth has complement pathway deficiencies. And the ninth is phenocopies of primary immunodeficiency. Now, this is a new category and a very interesting one where acquired autoantibodies can disrupt the immune system and present as a primary immunodeficiency, or somatic mutations may occur later in life, disrupting the immune system. So it's important to know that the ninth category are not necessarily inherited immune deficiencies, but they present like a genetic inherited immune deficiency would. So in this presentation, because the breadth of immune deficiencies is very large and continues to grow, we will touch upon categories 1, 3, and 5 under laboratory diagnosis, because as rare as they are, these are still the immune deficiencies that are seen more commonly in clinic. So let's move on to laboratory analysis of immune deficiency. Of course, laboratory analysis should be directed by the clinical presentation. And so it is important to take a good clinical history, including a family history. As I had mentioned previously, when the clinical presentation is very obvious, such as a baby boy presenting with very severe bacterial infections, it may guide the next step of laboratory diagnosis. In general, an initial immune assessment done in the laboratory would include a complete blood count, or a CBC, serum immunoglobulin levels, lymphocyte subsets, which would include broadly the T cells, B cells, natural killer cells, their percentages and absolute counts, and complement function. Complement function tests the integrity of the entire complement pathway, and it's known as the CH50 test. So this category of tests gives us a broad overview of how the immune system is working. So if all of these parameters are normal, and there is still a suspicion for immune deficiency, it's possible to go further into additional phenotyping of these immune system cells and their functional analyses. Some of the common tests are listed over here. We can look at T cell proliferation, or the ability of T cells to respond to mitogenic or antigenic stimulation. We can further subset T and B cells, look at regulatory T cell analyses. Particularly in dysregulated conditions, regulatory T cell analyses might be helpful. This is a small subset, less than 10% of T cells in the immune system that regulates the immune response of most of the other cells and keeps them in check. We can also look at neutrophil oxidative burst, which is an important test for the analysis of neutrophil function. So again, this is not an exhaustive list by any means. These are some examples of additional phenotype and function. It's important to understand that the initial immune assessment is offered generally by many clinical laboratories around the country. The additional phenotype and functional testing may be offered in some of the more specialized laboratories that focus on immunological testing. And finally, it's important to follow up with genetic analyses, because at the end of the day, once we do identify an immune deficiency, it's important to know what the mutation is. Is this inherited? In some cases, it's possible to speak with the family and determine if any other family members might be at risk. What is the risk for this child if eventually has a family to pass this mutation on to the next generation? So the genetic analysis is a key component of the laboratory analysis of immune deficiency. However, today we will be focusing mostly on the immunological, phenotypic, and functional analysis. So let's talk a little bit about the disorders in Group 1, and I will focus mostly on severe combined immune deficiency, otherwise known as SCID. So SCID is characterized by very severe defects of both the cellular and the humoral or antibody component of the immune system. The incidence is 1 in 100,000. However, if we look at specific populations, the incidence may be higher. So here's an example of the Artemis gene that is involved in the development of these immune cell populations. These mutations occur at a much higher rate in Navajo Americans. And so while we might say the global incidence is much lower when we do look at specific populations, particularly populations where there may be consanguinity, the incidence of some of these mutations can be much higher. So how do SCID patients present? They are at very high risk of serious infections, including opportunistic infections, and organisms of low pathogenicity that may not affect people with a normal immune system can cause serious infections in these particular patients. There's failure to thrive, and unless it's recognized and treated immediately, these babies can succumb to infection. So SCID has been recognized as a disease that, if recognized early in life, can be treated and managed well, and in many cases give these babies a better chance at a normal, healthy life in the long run. So newborn screening for SCID was started in 2008. Now, newborn screening for a number of metabolic diseases has been in place for many years, and what is used for these metabolic disorders is the blood spot on the Guthrie card. A heel prick is done when a baby is born, and blood is spotted onto this Guthrie card and used for newborn screening tests. So using this rationale in 2008, a molecular test was developed to screen for severe combined immune deficiency. Now this cartoon gives an idea of how this test is performed. The dried blood spot on the Guthrie card is used. A 3 millimeter punch is taken out of the card. The DNA is extracted, and the DNA is examined for integrity by using PCR to amplify beta-actin. So this is a housekeeping gene. If the levels of beta-actin are normal, the quality of the DNA is good. Along with beta-actin, a particular gene that's involved in development of the T cell receptor is also amplified, which is an indication of the numbers of newly developed alpha beta T cells. Now, this piece of DNA that's amplified is called a T cell receptor excision circle, or TREC, and shown here is the formation of this TREC, or T cell receptor excision circle. So when the TCR, or the T cell receptor, forms in these T cells that are within the thymus, the T cell germline DNA is rearranged using a number of different genes that are involved in the process. And as the TCR is rearranged, a portion of the DNA is pinched off and is left as a circle of DNA within the cell. It is not used for the T cell receptor. It simply is a circular piece of DNA called a T cell receptor excision circle that is present in very high numbers in these T cells that are exiting the thymus. And so shown in this cartoon is the germline T cell receptor locus above. On the left-hand side, you see the rearranged TCR locus containing portions of the VDJ genes. And on the right-hand side is a circular piece of DNA called the TREC, or the T cell receptor excision circle. So PCR primers can be used to amplify out this T cell receptor excision circle and quantified. And quantification of these TRECs tells us whether the baby has produced sufficient numbers of alpha beta T cells. Now, this test can be done a few days after birth, giving us an indication of whether the babies have produced adequate numbers of T cells. So what is done with that information? Now, before we move into that, the current state of newborn screening for SCID is presented here. This is from a paper in 2017. Newborn screening for SCID was started in 2008 by the state of Wisconsin and then adapted by California, and since then has spread across the country. And so as you can see from this map, almost all of the states across the United States have now implemented screening for SCID. Louisiana and Indiana are implementing it this year. And this is a huge achievement for progress into understanding that these genetic defects of the immune system, if recognized early on, can give these babies a chance at a better life as they are treated well within the first year of life. So what do we do following a SCID newborn screen for adequate numbers of T cells? This is the state of Colorado's workflow. Taking a look at the blue box above, when a baby is born and a test for SCID is run, if the number of TRECs, or T cell receptor excision circles, is greater or equal to 40, which is the state established cutoff, then the baby's immune system is considered normal. We move down to the green square. No further testing is required. If the TRECs are less than 40, then this is considered an abnormal screen, and the test is repeated in duplicate. If the retest has a result of greater than 40 TRECs, then we consider this to be a normal test and no further screening is required. However, if the TRECs are less than 40, this is considered to be an abnormal test, but at that point it is really important to look at the beta-actin level for the integrity of the DNA. If beta-actin is normal and TRECs are low, this is considered to be an abnormal screening for SCID, and the test is repeated a second time to confirm, as shown in the red square. If beta-actin is low, it indicates that perhaps the quality of the sample was not optimal, and we could call this an inconclusive test and go back and repeat the newborn screening test again. So if the TREC test is either positive or inconclusive, then we follow it up by immunological testing. The testing involves a complete blood count, a TBNK panel, which enumerates T cells, B cells, and natural killer cells from peripheral blood. And we also look at naive and memory T cell percentages, and we'll discuss this in a little more detail. It's important to note that not all failed SCID screens are indicative of severe combined immune deficiency. Some of these screens may fail because there is secondary lymphopenia. This is very common in preterm babies, sometimes due to lymphatic loss following cardiothoracic surgery, or DeGeorge syndrome, which is really an issue with lack of development or partial development of the thymus. And so T cells are, although they are present, they're unable to exit through the thymus to the periphery as would normally occur. So it's important to note that a SCID screen that is low does not always indicate severe combined immune deficiency. So let's take a look at the very basic TBNK flow phenotyping for SCID. Now this is a flow cytometry-based assay performed in a number of clinical labs. It's the first line for confirmation of a failed SCID screen. And shown here are the data from two flow cytometry tubes. The top plots are identified T cell subsets. The lower plots identify T, B, and natural killer cells. So whole blood is stained with a number of monoclonal antibodies that specifically identify lymphocytes, CD4, CD8, CD3 T cells, B cells using CD19, and natural killer cells using a combination of CD16 and 56. And these are all surface markers that are specific to these cell subsets. So if you take a look at the first two plots on the left, CD45, which is present on all nucleated cells, immune cells, is used to specifically identify lymphocyte populations that are shown in red. The lymphocyte populations can then be identified based on the expression of CD3, so that identifies T cells, and the T cells can be further subsetted into CD4 and CD8 T cells, as shown in the plot on the top row, right-hand side. Similarly, taking a look at the plots on the bottom row on the right-hand side, we can separate out non-T cells into CD19 B cells and natural killer cells. And so a very simple flow cytometry assay using about 50 microliters of blood allows us to enumerate all of these lymphoid cells in the periphery and look at the numbers and percentages. So how does this help us when we're faced with a newborn screen SCID result that is low or zero? So this is an example from a 4-day-old baby boy who had a TREC level of 0, and a confirmatory flow cytometry test was run. Now, as you can see in the plots, there are clearly populations that are missing. So, for example, the second plot from the left is lacking CD3, CD4 and CD8 T cells are completely absent, CD19 B cells are present, natural killer cells are present in the plot on the far right, but it's very evident that all T cell subsets are low, and those numbers are shown in red below in percentages. So a typical SCID has absent or very low numbers of T cells. Now here we're presenting them as absolute counts, so less than 300 cells per microliter. The other lab findings are an absent or very low T cell function. T cell function is commonly measured by taking peripheral blood cells and stimulating them with phytohemagglutinin, which is a mitogen and induces proliferation of T cells. So T cell responses in true SCID is very low, less than 10% of the normal control or normal range. These babies may have T cells, but they may be of maternal origin. And these maternal T cells show a memory phenotype, which we will discuss a little later. It's important to note that true SCID is an emergency because these babies that lack T cells are very, very highly susceptible to viral infections, fungal infections, and because they may be exposed in their natural environment within the family home, it has to be treated as an emergency and the baby put in a situation where they're shielded from infection and evaluated for a bone marrow transplant as soon as possible. So for typical SCID that has been identified by laboratory diagnosis and genetic diagnosis, in many cases, a bone marrow transplant, which effectively replaces the immune system, gives these babies a chance at a healthy life. So this is just a list of genes implicated in SCID. It is by no means an exhaustive list. These are the most common genes that are identified in SCID. As you can see here in this table, we classify them broadly into T minus, B plus SCID, which indicates T cells are absent, B cells are present, or SCID in which both T and B cells are absent, shown on the right-hand side. So some of the genes that are involved here, the T minus, B plus SCID, as you can see, many of these genes are related to T cell specific molecules or proteins. For instance, the CD3 delta, epsilon, and zeta are all involved with the CD3 glycoprotein that is specific to T cells. T and B, T minus, B minus SCID, the genes involved here are more broadly involved in the development of these lymphoid cells, and so their effect is broader on both categories of T and B cells. So we had mentioned the utility of looking at naive and memory T cell analyses, and why is it important in SCID? So naive and memory T cells can be identified by staining these cells by flow cytometry with CD45RA and RO. CD45RA and RO are isoforms of the CD45 glycoprotein. CD45RA is present on naive T cells. CD45RO is present preferentially on memory T cells. And as you can see in this flow cytometry plot on the left-hand side, it is a continuum, and they are not discrete populations. So there may be a preponderance of naive T cells or a preponderance of memory T cells, and there are some in the middle that might express both of these molecules. What we look at is the balance of these populations. As shown in the plot on the left, the pediatric cells tend to have a much larger population of naive T cells, whereas the adult sample has many more memory T cells. And this is what we would expect to see. In a newborn baby, we expect naive T cells to be close to 90%. So when we analyze naive and memory T cells in a newborn baby, and if we find that the phenotype is skewed towards memory, the following conditions can be considered. So leaky SCID, where there may be a few T cells that are in the periphery, but because there are only a few T cells, they may undergo what is called homeostatic proliferation, and they may respond to self-antigens. So they increase in number, develop a memory phenotype, and they may actually react against self-antigens. Omen syndrome is another case where there are reduced numbers of CD3 T cells, but they present with a memory phenotype. And then finally, as we've discussed earlier, maternal engraftment, or having some of the mother's T cells in the baby's system, we can certainly identify this by their memory phenotype as well as genetic studies. So when analyzing a sample from a baby with a failed SCID screen, it's important to not only look at the TB and NK populations, but also the naive and memory T cell populations. And both of these tests can be done very simply with small amounts of peripheral blood, usually about 50 microliters for each of these tests. So let's move on to antibody defects, or group 3 of the classification of primary immunodeficiencies. When we talk about antibody defects, we usually think about B cell development. And B cell development in this cartoon there are two stages to it. We can look at the stage of development that occurs in the bone marrow and defects of maturation that occur in the periphery. So development of B cells starts off from the hemopoietic stem cell, proceeds through the pro-B stage, the pre-B stage, and the immature B cell, which expresses IgM on its surface before it exits to the periphery. In the periphery, the mature B cell undergoes encounter with antigen. It matures. It can either take the form of a plasma cell and secrete antibody or become a long-lived memory B cell. And throughout this process, one of the important pieces is the ability of the B cell to switch from producing IgM to producing IgG or IgA or IgE. And so there are a number of genes involved both in development and in maturation. And depending on where mutations occur, we may either not see B cells in the periphery, as would be the case with defects in Bruton's tyrosine kinase or BTK, which we will discuss in a little more detail, or defects in some of the CVID-related genes that occur in the periphery can block the development of memory B cell populations and antibody-producing populations. So let's take a look at some of the prototypic defects in B cell development. Now, this is a case of a male with recurrent infections, had low or absent immunoglobulins in the periphery. Flow cytometry analysis was conducted on a peripheral blood sample. And as we see in the flow cytometry plot, we can identify lymphocytes in red on the upper left-hand side of the screen. Both CD4 shown in blue and CD8 shown in red are present. However, when we look at the plot below, we see adequate numbers of CD3 T cells in green, but no B cells. So this is a case of Bruton's tyrosine kinase mutations, or BTK, which lead to X-linked agammaglobulinemia, or XLA. So this is, again, an X-linked condition, so presents in boys or males. There's a history of recurrent infections, predominantly of the respiratory tract. These patients lack circulating B cells, very low immunoglobulins of all classes, and it's possible to confirm the BTK protein expression, presence or absence by flow cytometry. It's also expressed in monocytes, and it's possible to pick it up in monocytes when B cells are absent. So in a patient with BTK mutations, B cells are absent, monocytes do not express BTK protein, whereas a normal control will express the protein in monocytes and B cells. And of course, sequencing is always recommended to clinch the diagnosis. So BTK or XLA is the prototypic antibody deficiency where B cells are absent in the periphery. So we'll move on then to defects of maturation. Now in this case, B cells are present in the periphery. They can be detected by flow cytometry. However, they're not functioning in the proper way we would expect them to function. So one of the syndromes that are often encountered with antibody defects of maturation is the hyper-IgM syndrome. Clinical features here are increased susceptibility to infections. In this case, the patients have very high levels or sometimes normal levels of IgM, but absence of IgG and IgA. The most common form of hyper-IgM syndrome is also X-linked. There are other variations which are autosomal recessive, and these can present in either males or females. The hallmark of all of these syndromes is the inability of B cells to switch from making IgM to making the other classes of immunoglobulins. And so in this cartoon, one of the major defects we often find with hyper-IgM syndrome is a CD40 ligand defect. This is an X-linked defect. And interestingly, the defect occurs on the T cell, not necessarily the B cell. CD40 ligand on the T cell has to interact with CD40 on the B cell in order for the B cell to switch from making IgM to IgG, IgA, or IgE. So CD40 ligand is an X-linked disease. CD40 may be mutated, causing hyper-IgM syndrome, as can class-switching recombinase and a couple other genes along the signaling pathway, all of which are involved in the switching of the B cell from making IgM to making the other subclasses. So we'll take a look at some testing for hyper-IgM syndrome in general. Hyper-IgM syndrome due to any of these defects, CD40 ligand, CD40, or CSR, class-switch recombinase, all may present with a similar clinical picture. We have normal or elevated IgM, low IgG, low IgA, and increased susceptibility to infection. These patients will have B cells in the periphery as shown in this flow cytometry plot. If you look at the middle plot, we are identifying B cells here using CD20, and they are present. In the lower plot, we can further subset B cells into unswitched B cells that express IgD on their surface and switched B cells that lose IgD expression and make IgG, A, or E. And they all express this molecule CD27. So they're all memory B cells, some of which make IgG, A, or E, and some of which make IgM. So hyper-IgM patients have increased unswitched memory B cells that are making IgM, or in general, they may just have low memory B cells. So again, a very simple flow assay using a small amount of blood can tell us whether these memory populations are present in adequate percentages or not. So to get a little more specific and determine which of these defects is causing hyper-IgM syndrome in these patients, we can also analyze CD40 ligand expression on T cells. Now, the important thing with CD40 ligand is that it is seen only on T cells that are activated. So looking at the flow cytometry data on the left, we activate T cells in culture for a short period of time, about 4 hours, and then we identify CD4 T cells shown in green by their expression of CD3 and their lack of expression of CD8. Taking these CD4 T cells, we then look for specific expression of CD40 ligand, which is shown in these histogram plots below. The top one shows an isotype control, which doesn't show specific staining for CD40 ligand. The middle one is specific for CD40 ligand, and most importantly, we use an activation marker, CD69, to indicate that the T cells have activated. Now, this is important because if the T cells have not activated well in this test and CD40 ligand expression is absent, the test result is inconclusive. We need to know that the cells have been activated and do express CD40 ligand. So how would these results look in a patient with CD40 ligand mutations? As shown here in red is a schematic of a male child, elevated IgM, low IgG, low IgA, opportunistic infections, and as we see here, there is no CD40 ligand expression shown by this red histogram in the presence of good activation and CD69 expression. As this is an X-linked disorder, it's important to also analyze the mother's sample. And as shown in green, the mother has two peaks, one that is defective, no expression of CD40 ligand corresponding to the patient, and one that is normal corresponding to the healthy control sample. And this is an important piece of information. As an X-linked disorder, the mother has one normal X chromosome and one abnormal, and the male child has a 50-50 chance of inheriting either the mutated X chromosome or the normal X chromosome. So being able to analyze the mother sample and knowing that there is a carrier state allows advising the family in terms of the impact of these genetics on perhaps any other children. Daughters may have a chance of being a carrier, for instance. So we'll move on to group 5 and talk a little bit about congenital defects of phagocytes. So again, with neutrophils, we can consider defects of development that can be intrinsic, genetically acquired, or acquired defects that are due to, say, drug-induced neutropenia. Defects of function can affect multiple functions of the neutrophils, either their adherence to the endothelium, their ability to move or chemotaxis, their ability to produce good reactive oxygen intermediates that are responsible for bacterial clearance, or some of the extracellular mechanisms that they use to combat infection. Now, for the purpose of this talk, we will only focus on a couple of the defects of function that are more commonly seen in the clinic. We'll talk a little bit about defects of neutrophil adherence and defects of neutrophil oxidative burst. So just to go over defects of neutrophil function, this cartoon taken from a paper published several years ago nicely illustrates the functionality of neutrophils. Neutrophils found in the bloodstream tend to roll along until they receive a stimulus, perhaps from an infection, at which point they will arrest on the endothelium due to the expression of surface molecules called integrins. Once they arrest on the endothelium, they undergo something called diapedesis, where they squeeze through the endothelium to get into the tissue spaces. By the effect of chemotaxis, which is their ability to move towards a chemical stimulus, they move towards the site of infection. They phagocytose the organisms, and then they produce reactive oxygen intermediates to clear the infection. So along the way, a number of genes are involved in this process, as are illustrated in this cartoon. And we will talk a little bit about the integrins and mutations in these integrins that are important for adherence can lead to leukocyte adhesion defect, or LAD, LAD 1, 2, and 3. We will discuss LAD 1. And the genes involved in the oxidative burst or respiratory burst, that mutations in these genes can lead to chronic granulomatous disease. So we'll discuss the laboratory diagnoses for a couple of these conditions. So leukocyte adhesion defect, these mutations are characterized by recurrent severe infections with bacteria or fungi. One of the hallmarks of LAD is delayed separation of the umbilical cord in babies. And this is important to note because separation of the umbilical cord is a process of inflammation. We require neutrophils to migrate to the site. And by the process of an inflammatory reaction, the cord separates. In the case of LAD, the neutrophils cannot get to where they have to go. And so the umbilical cord separation is significantly delayed. Also significant is the absence of pus at the site of infection, again because neutrophils are unable to migrate to that site. So mutations in the CD18 gene affect expression of the integrins CD11A, B, and C. These integrins are present on the surface of the neutrophil, and their expression and function is important for neutrophils to tether to the endothelium and then to get into the tissue spaces to the site of infection. So how do we diagnose LAD1? This is done again by a very simple flow cytometry assay. We take whole blood as shown here. We choose to analyze just the neutrophils as shown in red on the top left. And then the specific expression of the integrins CD11A, CD11B, and CD11C, as shown in the histograms below, is examined. It's important to note that CD18 or the beta chain associates with each of these molecules so that they are expressed on the surface adequately. And we measure all four of them, CD11A, B, C, and CD18. So let's take a look at an LAD patient. Now this patient underwent bone marrow transplant, and as we can see here in the flow cytometry plot, the expression of CD11A, CD18, CD11B, and CD11C all show two peaks. The first peak is very similar to the isotype control, indicating that there is no expression of the integrins. The second peak, which is much smaller, is illustrative of the newly engrafted bone marrow and the fact that normal neutrophils are now developing that express these integrins. Although they're in much lower numbers, this assay can be used to monitor the patient over time and determine the extent of chimerism of the normal engrafted bone marrow and the defective population. So finally, we'll take a look at neutrophil oxidative burst. Now, neutrophil oxidative burst is the tail end of the neutrophil physiological process and clearance of infection. So at the end of this process, the neutrophils engulf the invading organism and release reactive oxygen intermediates to clear infection. So the way this occurs is through the NADPH complex. The NADPH complex is composed of two protein subunits that are present within the cell membrane, GP22 FOX and GP91 FOX. In the presence of infection, a number of cytosolic proteins are recruited to the membrane, GP47, 67, GP40, FOX, and RAC2, and together they form the NADPH complex that allows the electron transport chain to then release reactive oxygen intermediates that are mediators of antimicrobial activity. So mutations in any of these proteins can lead to chronic granulomatous disease, and the incidence is shown in this table. The GP91 FOX, or the major subunit in the membrane that's encoded by the gene CYBB, is inherited through an X-linked mechanism, and mutations in this gene account for about 2/3 of the incidence of chronic granulomatous disease that is seen in the United States. So this particular disease, the X-linked form, can present in baby boys with very severe infections, a very classic presentation. However, autosomal recessive mutations can occur in most of the other subunits, causing perhaps a milder disease. Just depends on the extent to which oxidative burst is affected. Now, these mutations, the incidence again is shown in this table. P47 at 20%, P22 and 67 at 5%, and P40 to date, one case has been reported. So to some extent, all of these genes, mutations can be inherited in an autosomal recessive manner, causing perhaps milder disease, and can be seen in either males or females. So the clinical presentation of X-linked chronic granulomatous disease is in males. They present with severe bacterial and fungal infections within the first 2 years of life, bacterial abscesses, very severe skin and gastrointestinal tract infections. The autosomal recessive form is males or females. They may have milder infections, really depending on the extent of the oxidative burst activity. And finally, it's important to note that with the X-linked disease, females are carriers and they may be asymptomatic, although some newer publications indicate that they may present with some autoimmune manifestations. So the laboratory diagnosis of CGD is again done in a number of labs around the country and is done with a fairly simple test in whole blood. So in this assay, whole blood is stimulated with PMA, and these cells are preloaded with a dye called dihydrorhodamine 1,2,3. Now this dye, once loaded into the neutrophils and the neutrophils are stimulated, can fluoresce very brightly because it gets oxidized by the reactive oxygen intermediates. So shown in this flow plot, on the left-hand side are neutrophils identified by the scatter gate. The second plot shows us the neutrophils with no dye. The middle plot is DHR loaded into the neutrophils, showing baseline fluorescence. And finally, the plot on the far right shows us the fluorescence of DHR once these cells are stimulated. And as you can see, the fluorescence is off scale here because it is so bright following stimulation of normal neutrophils. So we calculate an index value that is based on the fluorescence of the stimulated cells divided by the fluorescence of the unstimulated cells, and each lab establishes their own index value. In our lab, it is greater than 30, so an index value of greater than 30 indicates a normal oxidative burst. If it's less than that, then this is indicative of a defect in oxidative burst and needs to be investigated further. So let's take a look at some plots published in 2017 for chronic granulomatous disease. On the left-hand side, we see the top histograms showing a normal response. The resting cells show very little activity. The stimulated cells show a nice peak once oxidative burst occurs. X-linked chronic granulomatous disease in the middle from a baby boy show a complete lack of oxidative burst. The carrier phenotype is shown in the plots below. So the plot on the left-hand side shows no activity, and then there are two peaks following stimulation, indicating an abnormal and a normal neutrophil population. Autosomal recessive patients are rather more difficult to diagnose simply because there's more variation in their oxidative burst result. So shown here on the right-hand side are some plots from autosomal recessive CGD patients. One of the characteristics is a rather broad peak shown in the top plots on the right-hand side following stimulation. So instead of seeing a nice sharp peak upon stimulation, these patients may show a rather broader, flatter peak indicating perhaps compromised oxidative burst. It's also interesting to note that one of the patients in this publication showed no oxidative burst whatsoever, but were later confirmed to be autosomal recessive. When you look at the data, they look like a classic X-linked CGD. So again, it emphasizes that while the protein and functional studies are important, genetic sequencing is always important to confirm the diagnosis of primary immunodeficiency. So I'd like to leave you with some final thoughts. This is a very extensive topic, and I've covered a high level of primary immunodeficiencies and gone over some of the more commonly used diagnostic testing. Diagnosis of primary immunodeficiencies does pose challenges both to the physician and the laboratory. It may not follow a linear path. We may not be able to just go from TBNK analyses to B-cell phenotype or T-cell phenotype and function. These immune deficiencies may present in a very complex way, making it difficult to choose any one clinical test. For an obvious clinical presentation like chronic granulomatous disease, it may be easy enough to take the clinical presentation and go straight away to neutrophil oxidative burst. Laboratory analysis of phenotype and function may be followed by genetic analysis when required. And finally, I'd like to leave you with the thought that diagnosis of immune deficiency can be complicated because it may be influenced by other factors. So the types of infections that the patient has may in turn impact immune function. The genetics, the epigenetics, the environment, what are they exposed to? So it's very important to get a good combination of clinical history, family history, clinical examination, and careful laboratory evaluation to reach a diagnosis. I would like to acknowledge both of the institutions that I have worked at in the past 10 years. The Children's Hospital Colorado, where I am currently with the Department of Pathology, Laboratory Medicine, Flow Cytometry Lab, as well as the section of Clinical Immunology and Allergy within the Department of Pediatrics, and at National Jewish Health, where I was previously with the Advanced Diagnostics, Immunology, and Flow Cytometry Lab, and the Division of Clinical Immunology and Allergy, Department of Pediatrics as my collaborators. And of course, a lot of what I have discussed today would not be possible without the collaboration with the physicians and particularly the patients at both of these institutions. Much of what we have developed over the years is all because of patients coming in with these unusual presentations and requiring us to think beyond standard immunological testing and think about how best to go beyond that to more extensive phenotyping and function. So thank you very much for listening in, and I'm happy to take any questions.
Thank you, Dr. Knight, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask Question box located on the far left of your screen, and we'll answer as many of your questions as we have time for. So let's get started. Our first question is, if adequate numbers of T cells are detected by flow cytometry in a patient's peripheral blood sample, does it rule out immune deficiency?
Thank you, Alexis. That is a really important question. One of our, the first line of diagnosis, laboratory diagnostic tests is the TBNK analysis, which detects T cells, B cells, and NK cells. As we've seen in the presentation, when we take the example of a failed TREC screen and analyze a baby sample as a follow-up, we may actually detect T cells. So does it necessarily rule out immune deficiency? Not necessarily, because these T cells could be of maternal origin. They could be memory T cells. They could be functionally impaired. Some of the genes involved in T cell development do not impact T cell development, but impact T cell function. So when we are suspecting immune deficiency very strongly from a clinical perspective, and even if we do detect adequate numbers of T cells, if the suspicion is high enough, we should move on to further phenotyping them to see if they are naive or memory or subsets of memory T cells, and are they functional? If we stimulate them, will they proliferate in the laboratory setting? So follow-up testing is still very important.
Now, Dr. Knight, our next question. Is a failed newborn screen for SCID necessarily indicative of a genetic defect?
So a failed newborn screen for SCID indicates that there are low numbers of T cells. It is important to follow up with flow cytometry to determine if there truly are low numbers of T cells. It's not always necessary that these babies that fail the SCID screen have a genetic defect. Sometimes it could be due to a loss of lymph fluid, lymphatic leakage, so the T cells are not circulating in the periphery, but they are being generated in normal numbers. A failed SCID screen may be commonly seen in premature babies, so it's important to understand the other clinical features before going on to genetics. In case of a premature baby, testing may be warranted in a few weeks to allow for the immune system to mature and then repeat the test again. Again, another example that we touched upon is the DiGeorge syndrome, where the thymus may not have developed adequately or is absent. So again, T cells are being produced. Normally, this is not a genetic defect of T cell development, but it is instead a problem with the thymus. So not all failed SCID screens necessarily indicate a genetic defect.
And it looks like we have time for one more question. When you do an immune assessment of a patient, you mentioned running CBC, immunoglobulins, and a complement in addition to the T and B cell evaluations. Can you comment on whether you order all of these tests at the same time, or if results are of some dictate further testing?
That's a great question, Alexis. And yes, so we normally go with an initial screen. A complete blood cell count is almost routine when you're examining a patient with immune deficiency just to get a sense of numbers of lymphocytes, monocytes, and so on. Again, serum immunoglobulins is a good indicator. But again, we often go with the clinical picture. Now, if the patient has severe infections, say with capsulated bacteria, pneumococcal infections, we might lean more towards looking at the immunoglobulins and complement, because both of those components are really important for protection against capsulated bacteria. If the patient has, say, severe fungal infections or viral infections, we might lean more towards analyzing the T-cell arm of the immune system. So while this broad screen is helpful, I think in many cases, I would say the community setting, when you're thinking about immune deficiency, a CBC, immunoglobulins, TBNK at the very least, are very useful to get a global assessment of the immune system. If there's a strong suspicion or clinical history of encapsulated bacterial infections, then complement testing could be added in, since it is a critical component.
Thank you, Dr. Knight. Do you have any final comments for our audience?
I would just like to say that it's been a pleasure developing and putting this webinar together, and thank you to both LabRoots and Binding Site for hosting it. This primary immunodeficiency, I would say, is an area that is continually growing. New defects are continuously being identified. The laboratory plays a really important role in working collaboratively with the physician in helping to analyze, diagnose, and follow these patients. So again, thank you all for listening in, and I hope that you have found this webinar useful.
Thank you again, Dr. Knight, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Binding Site, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information that you provided at the time of registration. This webcast can be viewed on demand through May of 2019. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.